I'm Ebubechukwu Abolo, and this is my presentation on the likelihood of developing autism spectrum disorder as a result of sleep disruption during early life in autism spectrum disorder model mice. So, this presentation will be divided into sections. Section 1 would be the components of sleep through development. Section 2 would be synapses and autism. And section 3 would be the consequences of early life sleep loss. So firstly, sleep is a set period when the mind is put at ease and the brain may return to a homeostatic balance. Sleep dominates early life and continuously changes throughout one's lifetime. Though sleep has been linked to synaptic remodeling that takes place during early life, the role of sleep in relation to the brain's development is not yet clearly defined. So in the components of sleep through development, we have non-REM and then we have REM. So non-REM sleep, also known as slow wave sleep, is characterized by high amplitude and low frequency brainwaves during development. On the other hand, REM sleep is characterized by lower amplitudes because there is less neuronal synchrony, thus it resembles the waking state. So many studies have begun to link neuropsychiatric development disorders with early life sleep disruption. In a 2019 study showed that nearly 80% of preschoolers with autism experienced disrupted sleep, and infants sleep nearly 18 hours a day, while adults sleep 8 hours a day, with only 2 to 3 of those hours being REM. And so this drastic difference in sleep behavior and architecture has led scientists to question what the role of sleep is during development and if it is different between children and adults. Additionally, the effects of poor sleep in children with autism spectrum disorder are this worsening social skills, more severe repetitive behaviors, increased difficulty making friends than other children, and then lower scores on intelligence tests. Synaptic homeostasis hypothesis states that sleep encourages a decrease of synapses in response to an increase in synaptic activity occurring during periods of wakefulness. And this is a part of the synapses and autism portion of the presentation. So sleep and synapse regulation. Point A is that children with autism have more synapses than neurotypical children, and a surplus of synapses can affect the way in which the brain functions. The brain cells of children with autism, in a study done by Columbia University, found that brain cells of children with autism were filled with old and damaged parts and were very deficient in a pathway known as autophagy, which is just how it's just a pathway of synapse regulation in order to get rid of the damaged parts of synapses. And a possible solution to this lack in synaptic pruning is the drug rapamycin. And sleep contributes to the brain in language acquisition, auditory discrimination, visual learning, and motor development. And sleep deprivation during development is associated with long-lasting developmental consequences. Synaptopathies, which include abnormal synaptic morphology, number function, and exertory/inhibitory balance and expression of genes encoding synaptic proteins, have been associated with ASD. Thank you.